Oh, good. We've got construction happening. That's fun. Oh, that's lots of fun. Okay. Well, I have your tests. Do you want them today or do you want them on Wednesday? We go over with everybody.
I could look at them now.
You look at them. I could have given these to you like hours ago, and you could have been looking at them when I was messing around over here. So I did, this test was quite hard, and it was not hard intentionally. So I did do some magic on it. Basically, I threw out a couple of questions. I accepted a couple of answers on a couple of questions, even if they weren't like 100%, because I felt like a couple of them had like a best answer and an okay answer, which again was not intentional. So I also weighted the multiple choice less by, usually I make them worth 2 points. Instead, I made them worth 1 point, which gave them a little bit less weight compared to the short answer. And then at the end, I just flat out curved it by adding 10 points. So that brought the average up into like a low B, which is actually quite high for an average, but that's okay. But it was kind of my attempt to make up for this being hard because it was a bit harder than I would have preferred and a bit harder than I had planned. So if you want to take a look at that, if you have any questions, we'll go over it in a little more detail on Wednesday. For today, we're going to be going over, we're just going to do the lac operon today. And then we kind of already talked about some of the levels at which eukaryotic genomes are regulated. We'll just leave it at that and we'll move on to 13. And then if we have time, we'll move on to 18. We actually might start 18 today. So we'll go ahead and do that. On Wednesday, your lab report is due for GMO. And then I've forgotten the date, but coming up, there's an extra credit assignment due also.
This is on the day of the final.
Is it on the final? Okay,
Yeah,
that's fine. So we've got that coming up. And then next Wednesday will be a quiz. It'll be over probably 12 and 13, basically whatever we get through today. And then the remainder of the population genetics will be on the final. So the final will be 12, 13, 18, 12, 13, 18, and if we get to any of 14, we've got a little bit of 14 that we haven't covered kind of throughout the rest of the semester because 14 is all the biotech stuff. So a lot of that we cover in lab or in places where it makes sense to talk about it. So final will be 12, 13, 18, and 14, and I will give you an outline of the topics next week because next week is our last week.
can't wait.
Are you both graduating?
Yeah.
Are you graduating, DJ? Are you graduating?
I have to take a couple of classes.
Oh, okay. Do you get to walk though? Oh, okay. I think you could if you wanted to, but yeah, I know some people are like, nope, I don't need to. That's cool.
Everyone I know isn't walking. It's not fair.
You don't need them anyway. Yeah, I mean, I feel like it's kind of, I hear students all the time like, nope, not gonna do it. And others are super excited. It's okay.
Why not? They should be. After 2 years of blood, sweat, and tears, I need to do this for me.
Oh, then you should do that for you. Like, absolutely. Like, some students don't want to celebrate it. Some students do. I think some are like, well, I'm just going to go get my bachelor's anyway. But I feel like absolutely, if you want to do it, you do it. go for it. Faculty will be there anyway, whether you go or not. So I will not be there this year. I am off the hook for this year, but I've done the last 3, so I'm okay now. And actually, Kim told me, no, don't attend.
No.
Yeah, because it was technically, so we go in cycles where we have basically our evaluation cycles come up every 3 years. So the year we're on an evaluation cycle, we go. Well, I just had for the first 3 years, you have an evaluation every year. And so this year was my 3rd year, but she was like, we have too many faculty on this cycle, so please don't go. I was like, okay. Because I actually was like, I mean, I will, like I was planning on it. And she's like, no, please don't. It's okay. We don't need you. And I was like, oh, wow, that's fabulous. I like being not needed. So I will not be there, but Jessica and Rachel will be there. So
I'll get to see them.
maybe Mike. I can't remember who else. But anyway, let's go ahead and get started since it's 10 minutes after we're supposed to be started. All right, so we were talking about an operon. So remember that operons are a set of genes that are all controlled by the same promoter. So there's often 2 sets of genes that are working together in this pathway. We usually have the gene that or the set of genes that we are regulating. So for us today, we're going to be talking about the lac operon. And then there's another gene that is for whatever our regulator molecule is. So that regulator molecule can be an inducer. Remember, an inducer is going to turn on a gene. And a repressor is going to turn off a gene. So all of our genes are generally going to fall into 2 categories. Those that are under positive control and those that are negative control. From last week, do you all remember which one was which? Which one is positive control? Which one is negative control? Or like, I'm sorry, what are we doing if we are positively or negatively controlling a gene is a better way to say that.
Stimulating gene expression for positive control.
Very good. Okay, so positive control. This is one that is off that you want to turn on. And then negative is the opposite. This one is on and you want to turn it off. So our inducers are going to be the ones that turn on those genes under positive control. And our repressors are going to be those that turn off our genes that are under negative control. So we can do either one of these depending on the type of gene. Usually our gene is going to be one or the other. So it's going to be something that's normally off and we need to turn it on when we need it, or it's going to be on and we need to turn it off. So the same gene is not going to be under control of an inducer and a repressor. It will be one or the other. So we've got a promoter for that regulator, that inducer or repressor. And then we've also got a promoter for our operon. And then as part of our operon, we have this operator sequence. So the operator sequence is where your inducer or your repressor is going to bind. And we talked about a couple different ways that those could work. So I'm not going to go back to that at the moment. We're just going to go ahead and go into the lac operon. So the lac operon is what we would call a negative inducible operon. So this means that it is off, but we are going to use an inducer to turn it on. So what that means is we are having no expression of our gene. We were going to turn it on when lactose is present. And I'm actually going to have a little asterisk right there because that's not 100% true. And we'll talk about that in just a minute. So also part of our operon, we have a set of genes. In this image here, we've got A, B, and C. Once the gene is turned on, whether it's positive or negative control, once it's turned on, RNA polymerase is going to bind, move through, copy all of those genes into messenger RNA, and then each one's going to be translated. Usually these genes that are together in an operon are all related to one another. So very often they are all parts of our enzymes that are steps in a metabolic pathway, or somehow all involved in the same process. So for our lac operon, it's just kind of the official definitions there. For our lac operon, we are going to have this negative inducible operon for lactose metabolism. So in other words, to break down lactose in a cell. So we're going to use this as our example of how operons work in prokaryotes. The idea is similar for all of them. If we are looking at a repressible one, we're kind of doing opposite. We're expressing the repressor in order to control the expression. So just a little bit of background. E. coli's preferred carbon source is going to be glucose. So if there is ever glucose present in their growth media, that's what they're going to use first. If there are other sugars present, they'll use up all the glucose, and then they will switch over to using anything else that's present. So the goal of our lactose operon is to break down lactose when there is no glucose present. And the reason that E. coli are going to break down lactose is that when you break down lactose, it is a 2-sugar molecule or a disaccharide, and it's made up of glucose and galactose. And so if it can break down that lactose, it now has a glucose source. So it's just kind of an extra step to be able to use glucose. A second ago on the previous slide, I said that the lac operon was a negative inducible that was off, and we're going to turn it on. That was a little bit of a lie. There is always a low level of beta galactosidase, a low level of beta galactosidase in the cell. So beta galactosidase is the actual enzyme that is going to break down our lactose molecule. So we've got our glucose and our lactose bound together. That beta galactosidase is going to break that molecule in half. It's going to cleave this bond here between the 2 sugars there. So it's always expressed at a low level so that if glucose runs out, the E. coli can very quickly switch over to metabolizing something else. So this one, it is a negative, which means it's turned off, but it's not fully shut off. It's kind of like you just have a little trickle. You don't fully shut off your water, you just have a little trickle. That's kind of what's happening with this one. And it's just so that the cell can switch over really, really quickly if it needs to. So we have a couple of enzymes in this process that are all part of the lac operon. There are 3 genes in the lac operon. We're going to focus on 2. One is called permease. So permease acts as a little channel protein that brings in lactose into the cell. So permease, along with beta galactosidase, are both going to be expressed at really low levels so that the cell can bring in lactose if it needs to, and break it down if it needs to. But when there's no more glucose and lactose is present, the cell is going to ramp up the production of these two enzymes. Beta-galactosidase actually does two things, it can go two different ways with its processing. If lactose is high, it's going to convert lactose into its two sugars. So it's going to do the job that I just explained, break that lactose into galactose and glucose. If lactose levels are low, it's actually going to convert it into a different lactose form called allolactose. So allolactose is actually going to be our inducer. So that allolactose, when it is present, that's how the cell is going to know that we need to turn on this apparatus. So low levels of lactose, we actually get allolactose. High levels of lactose, we get our breaking down into glucose and galactose.
Yeah.
All right, so normally, this is turned off. So normally we have a repressor that is present so that beta-galactosidase is not produced. So if there's no lactose in the growth media or no lactose in the environment, this is going to be turned off by a repressor. So a repressor is going to bind to this operator so that RNA polymerase cannot bind. So normally, in a normal situation, when glucose is present, we have no reason to really express this gene other than those low levels. So their repressor is going to be present there, blocking RNA polymerase. So normally, we have a repressor protein blocking that operator. Last week, we talked about that sometimes the inducer will bind the repressor so the repressor can't bind the operator. So that's what's happening here. A second ago, I said that allolactose is going to act as the inducer. So when lactose is present, that beta-galactosidase, I'm going to abbreviate it as B-gal, just so I don't have to write it 100 times. When lactose is present, beta-galactosidase is going to convert lactose to allolactose, which in turn is going to bind the repressor. Yes.
So is that when it like has lactose, is the word, or when it doesn't?
Yes, yes, when there is lactose present, yes. So if lactose is present, initially it's going to get converted into allolactose. And then as allolactose accumulates, it's going to interfere with that repressor. And so now the repressor can't bind anymore. Let me see what I've got forward. Yep, there's a picture of it. So I'll show you in just a second. So on the previous picture of the operons, I said there were two different genes. There's the one, the regulator gene, the promoter with the regulator, and then there's the lac operon. So we've got two different systems here. One is for actually encoding the repressor. So we've got a promoter for the repressor gene. And then we've also got the promoter and the lac operon. So we're not going to talk too much about this promoter and repressor, the operon there. We're just going to say that it's present. The gene for that repressor is called the LAC-L, or I'm sorry, that's the LAC-I gene. The capital I. LAC-I gene. So that one is producing the repressor protein. So that one is always on because the default for E. coli is to prefer glucose. So we don't want to express that operon unless we need to. So it's only being expressed just a little tiny bit. Otherwise, the repressor is blocking most of the production. So if there's no lactose, there's no reason to produce the lactose operon. There's no allolactose interfering with that repressor. Then we've also got our actual lac operon. So there are 3 genes as part of this operon. So we kind of remind ourselves what we've got going on. We've got the promoter and the operator. So the operator kind of, I drew it like that because the operator sequence and the promoter sequence overlap a little bit because that repressor is actually blocking RNA polymerase that's binding at the promoter. And then after that, we've got our 3 genes and then a termination sequence or a terminator. So we've got the LAC-Z. That LAC-Z gene is going to actually produce that beta-galactosidase that either converts lactose into allolactose or into glucose and galactose. We've got, after that, the LAC-Y gene. So that LAC-Y gene is the permease gene. That's the protein channel that's actually going to let that lactose in. So the more lactose we have in the environment, the more allolactose we get. And then once the repressors are bound, then beta-galactosidase can start producing glucose and galactose, can start breaking down that lactose. And then the permease is going to allow more of that lactose in. There's one more gene, the LAC-A gene. This one, the function is kind of still under investigation. They kind of think that the LAC-A gene is acting as kind of a cleanup tool when permease lets in lactose and as part of this whole process of allolactose production, there are some components that are produced that are basically kind of toxic to the cell. And so they think that LAC-A gene is kind of involved in like basically cleaning up the extra kind of byproducts of this whole process. So you guys don't need to worry about the LAC-A gene for now. Just keep in mind that there's another gene that's getting promoted, still under investigation. All right, so over here, we've got the two different conditions for the LAC operon. So on the top, we've got that repressor protein. So this is going to be controlled by or produced with that LAC-I gene. So this is the one that's just always going to be on. So it's continuously producing. Once lactose accumulates in the cell, all of those repressors are going to be bound up. So as the LAC-I gene is producing repressors, allolactose is binding them to keep them off of the operator up there. So when there's no lactose, that repressor is just going to bind to the operator and then block the transcription of beta-galactosidase. A minute ago, DJ, you asked about binding of the repressor. So when there is lactose present in the cell, we're going to convert first that lactose into allolactose, and that's what's going on down here. So we've got beta-galactosidase converting lactose into allolactose. So that's this little blue bubble here. You can see in the repressor protein, there's an active site for binding the operator up here. And then there's also an active site for binding the inducer down here. So when that allolactose is present, it can bind to that repressor, change the active site that is binding to the operator and cause it to fall off. So it basically interferes with the ability of this regulator repressor protein to bind to the sequence. So then when that happens, we can get RNA polymerase, that's this guy here, that'll go through, transcribe that beta-galactosidase permease, and then the LAC-A, the transacetylase. Are there any questions on this one? So once the beta-galactosidase starts being expressed, then we've got glucose down here, and then glucose can go directly into cell respiration. So just kind of this extra step so that the bacterial cell can utilize different sugar sources. One more thing to talk about for the LAC operon, and that is what glucose's role is in all of this. So on a previous slide, I said there were 3 components to lactose metabolism. There is the regulator sequence, there's the LAC operon itself, and then there's a third component called the catabolite activator. So this component is basically what happens when glucose is present. So glucose is sort of the standard for all of our cells. Glucose is the preferred carbohydrate because it's the one that can go directly into cellular respiration without any other steps. So when glucose is low, or I'm sorry, when glucose is present, the cell's just going to preferentially use glucose. That LAC operon is going to be produced just in very low levels. And it is basically inhibited by this protein called the catabolite activator. So that catabolite activator kind of acts as a secondary repressor for this whole system. But when glucose is low, and lactose is high, this CAP protein or this catabolite activator is basically blocked from the, from being able to bind to the promoter region in the front of the lac operon. So this one kind of has like basically two levels of control. We've got control by the lactose system itself, beta-galactosidase and allolactose, but then we've also got this control, basically glucose control. When glucose is present, the glucose catabolite activator basically acts as sort of a secondary repressor on the system. So we get just a little bit of expression really until glucose happens to run out, which depending on the environment may or may not happen. So the important part is the first part we went through with the allolactose and the repressors. Any questions?
And the CAP protein, I just got wrong.
So the promoter.
The promoter.
Yeah, the promoter basically hold on. Did I say it backwards? I hope I didn't say it backwards. I'm a little worried I said it now. I said it backwards. So I think I might have said it backwards. So your CAP binding, when I think I said it backwards, when your glucose is high in the cell, we've still got a little bit of those LAC genes being expressed. And so in order to allow for that expression to happen, this CAP binding protein comes in and basically allows RNA polymerase to bind, even though we're trying to keep it turned off. So I think I actually explained it backwards. Okay. So that CAP protein is, I said it was blocking. It's not blocking. The CAP protein is actually helping RNA polymerase, even though it is normally turned off.
That's only when the glucose levels are high.
Mm-hmm. And then when the glucose levels get low, then the CAP protein no longer binds, and then you get your allolactose as your secondary activator. So you've got two positive controls here that are allowing the repressor to, or counteracting the effects of the repressor.
I have a question.
Jacob, yes, go ahead.
Is this, hi everyone, is this recorded? Can I go back and listen to this again? Or
Yes.
is it just, okay,
Yes,
awesome.
I'm recording this one.
So where do I find that at?
I will post it in Brightspace. So right now it will be immediately, it should be available if you go into the class teams. Oops. Let me see if I can bring it up real quick. If you go into can you see the team screen right now?
Unfortunately, I cannot. I just see your icon, your profile icon.
Oh, okay. So it's just the little bubble of me in the middle.
Yeah.
Okay. So on the left-hand side of the screen, if you click the Teams icon, there's this, it says genetics class. Nope, that's not it. Hold on. Let me find where it is. Oh gosh, I don't know. When you first, oh, all teams. There we go. When you're on the main screen, you should see all of your classes. Are you familiar with what I'm talking about there?
Not really. I'll probably have to, I can just come to like office hours tomorrow and you can show me.
Okay. Well, so it is available in Teams immediately, like as soon as I stop the recording, but then I usually post it directly in Brightspace as well. So it'll be available as a link in Brightspace later this evening.
Okay, awesome. Yeah, that
Just
works.
in the chapter 12, yeah.
Just for the beginning part that I missed, and then my phone unfortunately is dying, so I might miss some of the end as well. So as long as I just go back through and watch it again.
Yes, you can. Yep. And then after class, I'll like I have to upload it to YouTube and link it and everything, but it will be there. It'll be there later.
Okay, sounds good. Thanks.
Yep, no problem. All right. Are there any other questions for Lac Operon? No. Okay. Then we're going to be done with 12.
Nice.
And we're going to switch over to 13.
Small question.
Yeah.
So all the cap done is let's get the tablets.
Yes. Yeah. Yeah. The cap well, so it lets the cell express beta galactosidase at a low level. And then once glucose gets really low and lactose gets high, then CAP stops binding because it doesn't need to bind anymore because now we've got the inducer that's going to allow even greater transcription. All right, let's go over to 13. All right. All right, so we are not going to worry about the remainder of 12. I want to get moving on to 13 and 18. We won't cover all of 13 in a lot of detail. 13 is kind of an extension of chapter 11 when we were learning about translation. So you all had your packets, the gene expression packets, and in part of that packet, there was problems where you produced a mutation into your sequence and changed the protein by missense or nonsense, mutation, frame shift, whatever. So we're going to talk about those. We're going to talk a little bit about DNA repair. So transcription control or gene control is basically happening before. So we could have actually talked about regulation before we did transcription. The mutation chapter is kind of an extension of translation. So basically looking at what happens when we have a mutation, the effect that it has on the protein, and therefore the phenotype of the individual. And then we'll talk a little bit about repair mechanisms. So quickly switching gears. So a mutation, we talked about mutations several times already. Mutations are really just any change to a gene sequence. We usually talk about them in sort of a negative sense because from an evolutionary standpoint, organisms are well adapted to their environment. They may not be perfectly adapted, but they're going to be pretty well adapted to their environment based on just previous evolution. So if a mutation comes up, it can basically have one of three effects. It can be beneficial. It can be neutral, or it can be harmful. So since organisms are generally well adapted already, most of our mutations are probably going to fall into that harmful category. Neutral can mean neutral in the sense that it doesn't affect the protein, or it can be neutral in the sense that it happens in a non-coding region and so doesn't really affect any part of the phenotype. And then sometimes, of course, you'll get something beneficial, maybe beneficial in that particular environment and not in another environment. But we can, of course, have beneficial mutations as well. Mutations are helpful for evolution because it increases genetic variation. So if you think back to Biology 101, we've got genetic variation is one of our sort of like positive effects of sexual reproduction and just reproduction in general. For prokaryotes, genetic variation, because we don't have sexual reproduction, because we don't get combinations of genomes, for prokaryotes, mutation is really the only way that you're going to get genetic variation, unless by chance you have some of that horizontal gene transfer that we talked about, transduction, transformation, or conjugation. But besides those, genetic variation is going to come from mutations. For sexually reproducing organisms as well, this is the way that you get new genetic information. So mutation is the only way that you get new information. Otherwise, for sexual reproduction, when we talked about crossing over and independent assortment, fertilization of gametes itself, all of those are going to basically kind of shuffle the information that you already have, the allele or chromosome combinations you have. But mutation is going to give the actual brand new variation, new alleles. As far as humans go, many of our diseases and disorders are the result of mutations. So usually we think of them as sort of negative because usually diseases and disorders are in some way affecting the phenotype of an individual. Another way that we can use mutations is just for understanding biological processes. So we can look at the difference between the normal and the mutant. In order to determine phenotypes. So I think I already handed it out. You all should have a handout that has, it's got mice, that's got the lac operon, it's got mice for model organisms. It's also got Arabidopsis for model organism, which is the plant model organism, and then it's got some information about lac operon and types of mutations. But very often in our model organisms, we use mutations to do something called a knockout. So a knockout is basically a mutation that is induced in a gene, so specifically targeted to a gene in order to determine phenotypes. So for example, very often mice are used for knockout studies just to figure out what human genes are doing. Again, because mice are mammal model organisms. So if a gene is doing something in a mouse, it's probably doing something really similar in a human. And so if you can induce a mutation, disrupt the function, stop the function, you can see which function gets lost. So we can use mutations to our advantage as well. There are two categories of mutation, well, actually three, but two that we are going to be talking about. We've got our somatic mutations. So these are mutations that happen in our non-sex cells. These ones are important, but not quite so important for the study of genetics because mutations in our non-sex cells are not passed on to our offspring. In other words, they're not heritable. So usually when we're talking about somatic mutations, we're talking about those mutations that lead to cancer. So these are going to be, if we're talking about cancer, these are going to be your environmentally induced cancer. So things like skin cancer, lung cancer, where you get some sort of environmental exposure that brings about the cancer. You can also have heritable cancers or inherit the propensity or the predisposition for cancers. But like for example, skin cancer runs in my family. So the chances of the sun actually causing a mutation in me is much higher because my skin's really light, runs in my family. But that skin cancer would not necessarily be inherited by my children. If the say UV damage or whatever happens skin or it'll cause cancer for me, but because it's somatic, not being passed on. Then we've got our germline mutations. So these are the ones that occur in the gonad cells. So the mutations that occur during sperm and egg production. So these guys could potentially be environmental. Most of them are going to come up as errors in DNA replication. So you absolutely could have some sort of environmental exposure that causes a germline mutation. You can also have errors in DNA replication. These guys are important for genetics because these are passed on to offspring. And then third type of mutation, the somatic and germline mutations could be either a gene mutation or a chromosomal mutation. We talked about chromosomal mutations when we were talking about translocations, inversions, duplications, deletions, fragile sites. So we talked about all of those. We are going to be focusing on sequence mutations. And so basically mutations that are affecting a single gene, whereas your chromosome structure might be affecting multiple genes. So we're going to kind of focus on those sequence mutations. The image down at the bottom shows basically the difference between the somatic mutation. So if we have a mutation in a somatic cell, it's going to just spread to all of the sort of daughter cells from that particular mutant. But if we get it in the sperm or the eggs down here, then it's going to be passed on to all cells in the daughter. So somatic is going to be some cells in the same organism. Gametes are going to be all cells in our progeny. All right, so from our packet that you all did, you all are a little familiar with kind of the basic types of mutations. So we have got several different types, a little more in depth probably than you went through in Biology 101. So as far as single base or small number of base substitutions, we have transitions and transversions. So these are going to be converting one base to another. We also can have insertions and deletions. So these guys are usually the most disruptive because they are going to, because they're shifting that reading frame, they're shifting the triplet code. And so this is going to affect every amino acid after that insertion or deletion. Whereas with your substitutions, you may only affect one of them. And then we've got another type of mutation called expanding nucleotide repeats. So this is usually, say, like 2 to 3 nucleotides that occur as high copy number repeats. So you get lots and lots of repeats of a very small number of nucleotides. At the very beginning of the chapter, and the chapter image is a picture of Lou Gehrig's. So if you've heard of ALS or Lou Gehrig's disease, this is an expanding nucleotide repeat disorder. So we'll go through and talk about these guys in just a little bit more detail. The image on the next page kind of shows the difference between those. All right, so just to kind of show the difference between the two, or the four, if this is our original sequence up here at the top, substitution is going to affect one single nucleotide. So in this case, we're substituting a T for a C. That's just going to affect that particular codon. What that looks like depends on the nature of the substitution. So it depends on if it is substituting for a different amino acid, one with different chemical properties, a similar one, similar amino acid, or a stop codon. So depending on the nature of that substitution, we may have a big effect on our sequence. We may have no effect at all on the amino acid that gets put in. Insertion, you can see the frame shift for the insertion and deletion. At the same point, if we put an extra base pair in, then we've changed all of our reading frames all the way down. And so this is going to get cause the amino acids to shuffle and most likely have a strong effect on the phenotype of the individual. So we're going to start with substitutions. There are two types here. We can have a transition, which is basically where we just switch one purine for another purine or one pyrimidine for another pyrimidine. So down here we've got our transitions. Remember, your purines are adenine and guanine. And your pyrimidines are going to be your thymine, uracil also, but since we're talking about DNA here, uracil's not really important at the moment. Thymine and cytosine. So a transition is going to switch purine to purine. Pyrimidine is going to switch thymine to cytosine. This one is going to basically not affect the overall structure of our DNA. So when we're talking about DNA structure, we said that a pyrimidine always has to bind to a purine so that we get an even width all the way across. If you switch a purine to a purine, you're going to maintain that width. So these are a little bit hard for the cell to detect because one of the ways that the cell detects mutations is by looking at alterations in DNA structure. So if we don't affect the overall large-scale structure of our DNA, the cell may have trouble correcting it. A transversion is going to be a purine to a pyrimidine. So our opposite switch there. All right, so then we've got expanding nucleotide repeats. So if we look at this sequence here, we've got this GTC, GTC, GTC over and over again. This is kind of common in the human genome. We have some regions that are full of these repeats. And then we have some genes that are also have these repeats. The problem is that when DNA polymerase is working on replicating, it can kind of get tripped up, basically kind of keeping track of all of these repeats. And so what can happen is that as you get these repeats going on, DNA polymerase can start basically kind of losing track. And when it does, it can form these hairpin structures. So what happens then is if this sequence here sort of pulls away and wraps around and binds to itself, well, then we've shortened the length that appears to have been replicated already. So then DNA polymerase will come in and basically re-replicate that region. And so what happens is you end up expanding that region, and the more repeats there are, the more likely that you're going to continue to expand it. So what happens here is you get long chains of the same amino acid. So we're still affecting the protein structure, but we're just increasing the chain of amino acids here. All right, let's go ahead and do a break. It's time for a break, so we'll do that and then come back to this in just a minute.